Dr. Patrick Hoos, CEO of Moffitt Cancer Center. Really happy to be here at the ASCO 2023 annual meeting with Dr. Andrew Brohl. Thanks so much for meeting with us.
Yeah, thank you, Dr. Hu. Yeah,
and thanks
happy
for
to be here.
Presenting this interesting study. So let us know, it's in Merkel cell cancer. What is Merkel cell cancer?
Well, Merkel cell cancer, it's a rare skin cancer. It's unfortunately one of the most aggressive skin cancers that we deal with. So these patients get rapid progression of their tumors, high risk for metastatic spread. But on the positive side, we've seen good immunotherapy responses for a lot of these tumors. So we're trying to build off of that success and make the next generation of immunotherapies that we can help treat these patients better with. So this is one example that we think might be helpful for these patients that's in early testing, but have some very interesting early results that we're excited to present here.
What is the treatment?
So this is an intralesional product, and it's we call it a personalized vaccine in the sense that it's supposed to get the normal existing antigens within the tumors to be exposed to the body's immune system after we put in this bacterial protein. And so then the idea would that be this sort of helps spark the immune system, be an initiation event where the tumors can start releasing more antigens into the system so that the immune system recognizes it better. And then once you do that, you should, in theory at least, be able to re-challenge them with checkpoint inhibitors and get a better response, even if they might have failed that in the first place. And so far, we've seen, at least anecdotally, a few patients that have seemed to had exactly that response. They failed checkpoint inhibitor. We put them on this trial. They got this intratumoral vaccine approach. Then we retried them with the same class of therapy, the same checkpoint inhibitor class, PD-1 or PD-L1, and I've seen a number of very pronounced dramatic responses, even though they failed that the first time. And so we're very excited about this, even though it's still early, that we're seeing these responses already.
And you're injecting it in one of the lesions, but are you seeing the responses in other non-injected lesions?
Yeah, exactly. So we're injecting it just in one or several of the cutaneous lesions, and sometimes you'll see a response after the injection just in the tumor you inject. But after you re-challenge them with the checkpoint inhibitor, we're seeing these systemic responses, even in areas that we didn't inject. So hopefully it's an initiation event that can then propagate throughout the body if your body's able to react not only to the bacterial protein that you put in, but if that's, again, sort of an initiation event that lets the body start recognizing some of the pre-existing tumor antigens that must be getting exposed to along the way.
These are patients where the immune checkpoint inhibitor did not work, and then you give the injection, and then you try the same drug, and then it works on tumors throughout the body.
That's exactly right, yeah. Right. So we're thinking it's an immune priming event to take a tumor that didn't respond well to these standard care therapies and hopefully can reprime the immune system to start recognizing or making these tumors more hot. And then if your body's immune system can recognize one of them that you injected or several of them that you injected, then hopefully that'll spread throughout the body. And at least in a few cases, it seems to be doing just that. So it's very exciting, early work, but that we're seeing these types of responses already, it's very encouraging.
Why do you think Merkel cell is immunogenic, meaning it can stimulate the body's immune system? Do you think it's because it's associated with a virus, the carcinoma virus, right?
Yeah.
And is that the antigen, or is there a lot of mutation burden in Merkel cell?
Well, it turns out that Merkel has two very distinct biologies from a molecular standpoint. One is a viral-driven tumor that has very few other antigens, and most of the antigens come from a virus. And then you have this other set of Merkel that is zero virus, but has a ton of neoantigens from the UV mutation, just like melanoma, but even higher. And so these tumors have either very immunogenic viral antigens or a large number of potentially immunogenic UV mutation antigens.
That's interesting.
And so you got one of the two, but not both. Usually.
Is it like 50-50, or what do you see?
There's a little bit more of the viral-driven ones overall, if you look at the literature. But if you get to the metastatic point, the UV-driven ones are a little bit more likely to get metastatic. So it gets closer to 50-50. And also in our demographic in Florida, it's closer to 50-50 because we have more of the sun-exposed individuals.
Do both subsets respond to immunotherapy in the same way?
Both have a very similar response. It's not like statistically different in any of the studies. And some people say there might be a marginal better response in the UV ones, but it's pretty close, and they both have a pretty good chance to respond in both cases.
Great. So what was your response rate?
Well, in terms of the re-challenge response rate, we saw 5 out of 7 total, or 71%, that went through this period where they had previously failed checkpoint inhibitors, got this therapy, and then were re-challenged with standard care afterwards and had this good response. So that's, again, the response rate to the re-challenge, not necessarily to the drug itself, but,
wonderful.
this prime effect, but a very, impressive for this
you can make a drug that wasn't working work. That's very important.
Especially a checkpoint inhibitor that can have a durable response like these days.
So what's the next step?
Well, we're expanding this trial to get additional numbers to support these results so far and get some additional biomarkers. But we're already in talks with the with the pharma sponsor and the FDA to try to move it forward in Merkel as hopefully a registrational trial, randomized trial is the next step. They're very encouraged by this and enthusiastic to try to move it forward. This is their lead compound, and we're working closely with them to be the lead site for that.
Well, very exciting.
Yeah.
Well, thank you, Dr. Andrew Brohl from our sarcoma and Merkel cell group at Moffitt Cancer Center.
All right, thank you.